Hello everyone. So today I'm going to talk about a very important topic, glycolysis, in certain detail. So glycolysis is important for bachelors, masters, as well as for getting a JRF position, anywhere for life science. So let's start this very important topic. So let's start with the introduction. Glycolysis occurs in the cytosol, right? So if I draw a cell here, and this is the nucleus which contains the DNA, and here is a mitochondria, right? So glycolysis occurs in the cytosol, right? And while the citric acid cycles occurs in the mitochondrial matrix. So mitochondria has two membrane structure, right? So within the matrix, the central area, here citric acid cycle occurs. Oxidative phosphorylation occurs in the inner mitochondrial membrane, which is here, the inner mitochondrial membrane. Here is where the oxidative phosphorylation occurs. Now, what is glycolysis? Glycolysis is the type of incomplete oxidation where 1 mole of glucose is broken down into 2 moles of pyruvate. And in that process, it generates energy in the form of ATP and NADPH, right? So, what happens in glycolysis is that 1 mole of glucose is converted into 2 moles of pyruvate, right? And in that process, ATP, 2 ATP and 2 NADH is produced as energy. So this is the whole idea of the glycolysis for the production of energy. So let's go into detail. So here you can see the whole thing in a chart form. So what is happening? Glucose is being converted into glucose 6-phosphate, which means a phosphate is being used. Where the phosphate is coming from? From the molecule of ATP. ATP is giving one of its phosphates to glucose and making it glucose 6-phosphate, and itself is becoming ADP, right? Then the second step for this is happening through the enzyme hexokinase. So this is a kind of phosphorylation is occurring where glucose is being converted to glucose 6-phosphate through the ATP, which is giving one of its phosphates to become ADP itself. The enzyme used here is hexokinase. Now another thing important here is that, as you can see, this is a non-reversible reaction. Glucose being converted to glucose 6-phosphate is a non-reversible, only one directional reaction. Now the second step, as you can see, glucose 6-phosphate is being converted into fructose 6-phosphate, right? The enzyme being used here is phosphoglucomutase or phosphoglucoisomerase. So an isomer is being formed where glucose 6, only the position of the carbonyl oxygen is being changed here. So glucose 6-phosphate is being converted into fructose 6-phosphate, right? Which is a reversible reaction. Now the third step is where the fructose 6-phosphate is being converted to fructose 1,6-bisphosphate. So as you can see, another phosphate is being added to the molecule. Now where the phosphate is coming from? It is coming from, once again, from an ATP, which is giving one of its phosphates to become ADP and giving its phosphate to fructose for it to become fructose 1,6-bisphosphate. The enzyme used here is phosphofructokinase. Now one of the important points here is that kinase is generally an enzyme which is used for phosphorylation, for addition or removal of phosphate, right? So kinase, wherever you see an enzyme that is named kinase, it is generally for the addition of or for the phosphorylation of a molecule. That's where a kinase enzyme is used. Now the next step is where the fructose 1,6-bisphosphate is being converted or broken down into two molecules. One is dihydroxyacetone phosphate DHAP and the other is glyceraldehyde 3-phosphate. So fructose 1,6-bisphosphate, here glucose was a 6-carbon molecule, glucose 6-phosphate was also 6-carbon molecule, fructose 6-phosphate was also 6-carbon molecule, fructose 1,6-bisphosphate was also 6-carbon molecule, and now this 6-carbon molecule is being converted or broken down into two 3-carbon molecules. The dihydroxyacetone phosphate is a 3-carbon molecule and the other glyceraldehyde 3-phosphate was also a 3-carbon molecule. So the 6-carbon molecule, which is fructose 1,6-bisphosphate, is being broken down into two 3-carbon molecules. Right. Now dihydroxyacetone phosphate cannot undergo the further processing. So what actually happens is that dihydroxyacetone phosphate is reversibly being converted into glyceraldehyde 3-phosphate, right? So at the end what we get is two molecules of this dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase. Right, so by the isomerization process this molecule is converted to this molecule. So at the end there are two molecules of this, right? So there are two 3-carbon molecule glyceraldehyde 3-phosphate. Now 2-glyceraldehyde 3-phosphate then undergoes another reaction where glyceraldehyde 3-phosphate dehydrogenase is then converted into 1,3-bisphosphoglycerate molecule, right? So 1,3-bisphosphoglycerate is formed through this enzyme glyceraldehyde 3-phosphate dehydrogenase, and here two energy molecules 2-NADH is produced. This is an important step. Okay, so in this process 2-NADH is produced from 2-NAD plus, right? So energy is being produced now. From here this is called an payoff phase. From here to here this is preparatory phase where preparations are being made, and from here we are getting paid off, which means energy is being produced. So as you can see in this step NADH, which is an high energy molecule, is being produced, right? So let's go to the next step where 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate molecule. Now again there were two glyceraldehyde 3-phosphate molecules, so from here all the molecules are 2 in number. Two 1,3-bisphosphoglycerate are made and then two 3-phosphoglycerate molecules will be formed by the enzyme phosphoglycerate kinase. Again here is a kinase, which means phosphorylation is being taken place, right? So what is happening, there are, as you can see from the name, there were two phosphates now, right? There were two phosphates in these molecules, but one of the phosphates which is from the first position is now given to an ADP molecule to make an ATP from it, right? So now ADP is getting an phosphate and being converted to ATP. Now because there were two molecules of 1,3-bisphosphoglycerate, so two molecules of ADP are being converted into two molecules of ATP because two phosphates are being given. Now the next step is where 3-phosphoglycerate is converted into 2-phosphoglycerate, which is of course an isomerization reaction which is occurring through phosphoglyceromutase enzyme, right? This is simply an isomerization process. The next step is conversion of 2-phosphoglycerate into phosphoenol pyruvate by the removal of 2 water molecules, right? This occurs through the enzyme enolase. Now phosphoenol pyruvate is then converted into pyruvate 2 2. So 2-phosphoenol pyruvate is converted into 2 pyruvate molecules through the enzyme pyruvate kinase. And now as you can see again that there is a kinase enzyme, which means something is being phosphorylated. So what is happening, that as you can see, phosphoenol pyruvate, and here is just pyruvate. So phosphate is again being removed from this molecule and given to 2 ADP molecules which are being phosphorylated to be converted into high energy molecules to ATP molecules, right? So this is the whole glycolysis process. Now we will go through each step in detail and slowly, knowing about every enzyme on the way. So step 1 is phosphorylation. This is the step 1 where glucose is being converted into glucose 6-phosphate by the enzyme hexokinase, right? And what happens for the phosphorylation of glucose into glucose 6-phosphate, 1 ATP is required, right? So glucose is phosphorylated by ATP to form glucose 6-phosphate, as you can see, right? Glucose is phosphorylated by ATP to form glucose 6-phosphate. Now the negative charge prevents the molecules from leaving the cell through plasma membrane. So what is happening, that because phosphate is being added to glucose, a negative charge is being given to this glucose by the addition of phosphate. Hence, due to this negative charge, the glucose will be no longer able to cross the plasma membrane on its own. For example, if here was glucose in the cell, generally through an enzyme or through facilitated diffusion, the glucose can cross the plasma membrane. Not on its own, it would require some proteins, some transportable proteins, but it could still cross the plasma membrane. But once it has been phosphorylated and a negative charge has been added to glucose, because it's charged and a large molecule, it could no longer cross the plasma membrane, and now it is trapped into the cytosol. So the next step of the glycolysis could occur within the cytoplasm. Now this is an irreversible step which occurs through the enzyme of hexokinase, right? So as you can see, this is a one-way process, a one-way reaction. It is irreversible and it occurs with the help of hexokinase enzyme, which requires Mg2 plus or Mn2 plus as its cofactor. The enzyme requires either this or this ion as a cofactor for it to work. Now another important thing about hexokinase is that the hepatocytes in the liver and the beta cells of pancreas have glucokinase, an enzyme named glucokinase, which is an isozyme of the hexokinase. Isozyme means that it is slightly different, but it still does the same function. So in hepatocyte and beta cell of pancreas, there is the enzyme called glucokinase instead of hexokinase. So what glucokinase does different from hexokinase, glucokinase, is that it has higher Km and higher Vmax value, which you can study through the Michaelis-Menten equation, which I can explain in later videos. So now let's move to the next step. The second step of glycolysis is isomerization. So phosphoglucoisomerase enzyme rearrange the carbonyl oxygen and convert it from aldose to ketose molecule. As you can see, this is an aldose molecule, glucose 6-phosphate, which would look something like this. right, glucose, while fructose looks something like this, right. So it is converted from an aldose molecule to a ketose molecule, right. Only the carbonyl oxygen rearrangement occurs, as you can see. So it is a kind of isomerization reaction, and this occurs through the enzyme called phosphoglucomatase, or what is also known as phosphoglucoisomerase, same thing, right. So now the glucose 6 phosphate is converted into fructose 6 phosphate. Now let's move on to the next slide, which is also the next step of glycolysis. Step 3 is phosphorylation. So in step 3, fructose 6 phosphate is now converted into fructose 1,6 bisphosphate by the enzyme phosphofructokinase, right. Again there is the word kinase in the enzyme's name, which means that phosphorylation is occurring. You can also see here that there is only one phosphate in this molecule, while there is fructose 1,6 bisphosphate. So now there was only one phosphate on the position 6. Now there are two phosphate, one on position 1 and another on position 6 in the fructose molecule. So fructose 6 phosphate is converted to fructose 1,6 bisphosphate by the phosphorylation of ATP, and ATP gives one of its phosphate to the fructose molecule and produces a fructose 1,6 bisphosphate. This is an reversible process which occurs by the help of enzyme phosphofructose kinase 1. Let's move on to the next slide. Step 4 is cleavage. Cleavage means breaking down. Now as you can see, fructose 1,6 bisphosphate is cleaved to produce two molecules of 3 carbon each. Fructose 1,6 bisphosphate is a 6 carbon molecule which is cleaved or broken down into two 3 carbon molecules, right. The first molecule is dihydroxyacetone phosphate and the second one is glyceraldehyde 3 phosphate, both of which are 3 carbon molecule compounds, right. It occurs through the enzyme called aldolase. Now first the glyceraldehyde 3 phosphate and second is dihydroxyacetone phosphate. These are the two molecules which are both 3 carbon, and this reaction is catalyzed by the enzyme aldolase. Now in step 5 there is another isomerization reaction. What happens in this isomerization is that like I said before, there are two 3 carbon molecules produced, dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. But in this process, in step 5, dihydroxyacetone phosphate is converted into glyceraldehyde 3 phosphate. So this molecule is converted into glyceraldehyde 3 phosphate for further reactions to occur, right. And this occurs through the enzyme triose phosphate isomerase enzyme, right. So the dihydroxyacetone phosphate is rapidly and reversibly converted into glyceraldehyde 3 phosphate by the enzyme triose phosphate isomerase. Let's move on to the next step. Now up until this step, until the 5th step, it was preparatory phase. From step 6 it is payoff phase, which means that now we are going to be paid off in the form of ATP or other high energy producing molecules, right. So this is the payoff phase where energy molecules are going to be produced. So in step 6 what is happening is that glyceraldehyde 3 phosphate is converted into 1,3 bisphosphoglycerate. So as you can see, glyceraldehyde 3 phosphate has only one phosphate molecule only one phosphate molecule, while here the 1,3 bisphosphoglycerate had two phosphates. So one phosphate is being added, which is not through ATP, okay, it is through an inorganic compound, but it is not through ATP. Now the glyceraldehyde 3 phosphate dehydrogenase enzyme converts 2 NAD plus into 2 NADH, NADH are high energy molecules which are equal to actually 2.5 ATP energy, right? One NADH molecules can give energy equal to 2.5 ATP, right? So here energy is being produced in the form of NADH, two NADH molecules, because there are two glyceraldehyde phosphate, so there are going to be produced 2,1,3 bisphosphoglycerate, and there is also going to be 2 NADH molecule, and no ATP is being used in phosphorylation, right? So NAD plus is being converted to NADH because of the oxidation of glyceraldehyde 3 phosphate, and the enzyme required is glyceraldehyde 3 phosphate dehydrogenase. This step is inhibited. This enzyme could be inhibited by a molecule called iodoacetate. Iodoacetate could inhibit this enzyme glyceraldehyde 3 phosphate dehydrogenase, so this step could be stopped or inhibited through this molecule. Let's move on to the next slide. Step 7. In step 7, substrate level phosphorylation occurs, which means that now ATP will be generated. In this step, ATP generation occurs, right? So substrate level phosphorylation happens when 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. So there are two phosphates in this molecule, which is then one phosphate is being taken out and from each molecule. So because there are two bisphosphoglycerate molecule, so 2 ADP, 2 phosphate will be taken out, and so 2 ADP will be converted into 2 ATP. 2 ATP will be generated through the enzyme phosphoglycerate kinase, and 3 phosphoglycerate, 2 molecules of 3 phosphoglycerate will be generated. Now there is a special case for this in the erythrocytes. Erythrocytes are also known as RBCs. So in the red blood cells, a glycolytic bypass is there in RBCs, where the production of 2,3-bisphosphoglycerate, that is 2,3-BPG, is increased. This process is known as rapport lumbering shunt. This shunt bypasses the phosphoglycerate kinase step and accounts for the synthesis and regulation of 2,3-BPG level that decreases the hemoglobin affinity for oxygen. So is the thing, within RBCs, this molecule, which is 1,3-BPG, sorry, 2,3-BPG, this molecule is important within the RBCs. What it actually does is that 2,3-BPG, BPG decreases the oxygen affinity for hemoglobin. As you know, RBCs carry hemoglobin, which combines with oxygen to carry it throughout the body. Now the function of BPG is to reduce the affinity of oxygen for the hemoglobin in RBCs. So it has an important function within the RBCs. Hence, within the RBCs, this enzyme is generally inhibited and a rapport lumbering shunt process takes place, through which production of this molecule is increased, right? So this was a special case within the RBCs. Let's move on to the next step, the step 8, where the 3-phosphoglycerate is converted into 2-phosphoglycerate by the enzyme phosphoglycerate mutase. So this is simply an isomerization reaction again, where 3-phosphoglycerate, the phosphate position is changed, and 3-phosphoglycerate is converted to 2-phosphoglycerate by the enzyme phosphoglyceromutase or phosphoglyceroisomerase, right? Same thing. Step 9. So in step 9, the removal of 2 water molecules. Again, you have to remember that here all the molecules has 3 carbon, and so 2 molecules of each, right? 2 molecules of 2-phosphoglycerate is converted into phosphoenol pyruvate by the removal of two water molecules, and this occurs through the help of enolase enzyme. This was step 9, which was a irreversible process. Now step 10, or the final step, is where phosphoenol pyruvate is converted into pyruvate. Two molecules of phosphoenol pyruvate is converted to two molecules of pyruvate, which is again both are 3 carbon molecules, right? This occurs through the enzyme pyruvate kinase, and as you can see, one phosphate is being removed. Phosphate, here phosphoenol pyruvate, this is just pyruvate. So phosphate is being removed, 2 phosphate, because there are 2 molecules of phosphoenol pyruvate. So 2 ADP are being converted to 2 ATP, right? So again, this pyruvate kinase enzyme requires Mg2 plus, K plus or Mn2 plus for the reaction to take place, right? So this was the whole glycolysis process in detail. And there are certain other things that one must remember. For example, here you can see in this picture that there is taking place a net reaction. So you have to see a net reaction, what is being produced and what is being used up. So as you can see in the first step, there is use of 1 ATP. So the things that are being used, I am putting negative in front of it. So 1 ATP is used here in the first step, right? The second is here another ATP is used, so again minus 1 ATP, right? The next step is 2 NADH is being produced, so plus 2 NADH is being produced. In the next step, 2 ATP is being produced, so plus 2 ATP, right? In the last step, again 2 ATP is being produced, so 2 plus 2 ATP. So if you see the whole, there is minus 2 ATP plus 4 ATP plus 2 NADH, right? So this is the whole reaction. Now minus 2 ATP plus 4 ATP, which equals to 2 ATP in total, plus 2 NADH. So 1 NADH is equals to 2.5 ATP. Now 2 NADH will be equal to 2.5 into 2, which is equal to 5. So 2 ATP plus, instead of NADH we are going to put 5 ATP. So in total 7 ATP is being produced in one cycle of glycolysis, where 1 mole of glucose is being converted into 2 mole of pyruvate and 7 ATP energy is being produced. So this is the net reaction. Now regulation of glycolysis. So because there are so many enzymes involved in this, but not all the enzymes within the process actually regulates the pathway or regulates the reaction rate, right? So which are the enzymes that actually regulates the reaction rate? Let's see. So allosteric enzymes, which are generally those enzymes which function as non-reversible reaction, right? So all those reversible reaction like this, these are generally not allosteric reactions, right? So let's see which are the enzymes which actually regulates the rate of glycolysis. So those enzymes are hexokinase, phosphofructokinase 1 and pyruvate kinase. So hexokinase, phosphofructokinase and pyruvate kinase. So kinase enzymes are generally those that is regulating the whole process. Now here we are using a term allosteric. Allosteric regulation means that every enzyme is having two sites. A site where substrate binds, substrate binding site, and another site which is allosteric site. So allosteric regulation occurs when the inhibitor or the activator binds to the allosteric site and not on the substrate binding site of the enzyme. So these three enzymes, the hexokinase, the phosphofructokinase 1 and the pyruvate kinase, are all allosteric enzymes. So glucose 6-phosphate inhibits the hexokinase. So what inhibits hexokinase enzyme? This glucose 6-phosphate is the molecule that inhibits hexokinase. Why? This is a kind of what you'd call a negative feedback loop, right? A negative feedback loop is being produced if there is very high level of glucose 6-phosphate, which would inhibit the hexokinase, which would in turn inhibit the whole glycolysis process, right? So this is the first. The second is that increased level of AMP concentration activates phosphofructokinase and pyruvate kinase. So phosphofructokinase and pyruvate kinase are activated by increased level of AMP, while increased level of ATP inhibits these two enzymes. Phosphofructokinase and pyruvate kinase are activated by increased level of AMP and inhibited by the increased level of ATP. So that's all for the regulation and the whole process of glycolysis. Thank you very much for joining in this video. Please like and subscribe.